Today we are going to discuss about digestion and absorption of lipid. Before proceeding to digestion and absorption of lipid, you should know what are the lipids. Lipids are heterogeneous organic substances insoluble in polar solvents like water, soluble in non-polar solvents like benzene, chloroform, ether and alcohol, actually or potentially related to fatty acid, and should be utilized by living organism. Any substance to be considered as a lipid should satisfy these 5 criteria. When you compare lipids with carbohydrate and protein, lipids are smaller molecules, whereas carbohydrate and proteins, these are larger polymers. Now we'll come to digestion of lipid. Just like carbohydrate and protein, lipids are also undergo breakdown into smaller components. But digestion of lipid possesses a special problem because of insolubility of lipid in water or aqueous medium present in gastrointestinal tract. As lipids are insoluble in water, they are not easily accessible to the digestive enzyme for its action. When digestive enzyme acts on lipid and form hydrolytic products, these products also tend to get aggregate to larger complexes that make poor contact with the cell surface, and because of that lipids are not easily absorbed. So problem is there in both digestion and absorption. This problem is solved by certain changes in the physical state of lipids which are connected to the chemical changes. This problem is mainly solved by a process known as emulsification. What is emulsification? It is a process of dispersion of lipids into smaller droplets by reducing surface tension, which occurs by the action of bile salts present in bile. This slide shows general overview of digestion of lipid. Digestion of lipid begins in mouth by the secretion of enzyme lingual lipase by salivary gland. This lingual lipase enzyme is having optimum pH, that is about 2.5 to 5. Lingual lipase enzyme causes breakdown of triacylglycerol, which is made up of short chain and medium chain fatty acid. Now what are the short chain and medium chain fatty acids? Short chain fatty acids are the fatty acids having 2 to 6 carbon in its structure, whereas medium chain fatty acids are the fatty acids having 8 to 14 carbon atom in its structure. Examples of short chain fatty acids are acetic acid, propionic acid, butyric acid, valeric acid and caproic acid, whereas examples of medium chain fatty acids are caprylic acid, capric acid, lauric acid and myristic acid. These short chain and medium chain fatty acids undergo release from the triacylglycerol by the action of lingual lipase enzyme. But in mouth, lipid digestion occurs very little because food stays in mouth for shorter duration. After mouth, when food enters in stomach, most of the fat is intact. Here it undergo mixing and breakdown into droplet. This fat undergo breakdown by the action of another enzyme in the stomach, which is gastric lipase. This gastric lipase enzyme is secreted from the chief cells of the gastric mucosa. Just like mouth, in stomach also fat digestion occurs in lesser amount. After stomach, when food enters into the small intestine, there leads to the secretion of two hormones, cholecystokinin pancreozymin and secretin from the duodenal mucosal cells. These two hormones causes contraction of the gallbladder and release of bile into the intestine. These two hormones also act on exocrine portion of the pancreas and causes release of enzyme pancreatic lipase, cholesterol ester hydrolase and phospholipase A2. The bile secretion, which is released from the gallbladder and which comes in intestine, causes breakdown of fat into smaller droplet, and later on these lipid digesting enzymes acts on its various substrate and produces the various product. The product which are formed are transported from the intestinal lumen to the intestinal mucosal surface by micelle. You can see here digestion of triglyceride, cholesterol ester and phospholipid. Triglyceride undergo digestion by pancreatic lipase to form two molecules of fatty acid and one molecule of monoglyceride. Here, fatty acids are released from the first and third carbon position of glycerol. Cholesterol ester undergo hydrolysis by the action of enzyme cholesterol ester hydrolase to form fatty acid and cholesterol. Phospholipase A2 enzyme acts on phospholipid and form lysolecithin and fatty acid. The peculiarity of phospholipase A2 is that it releases fatty acid from the second carbon position of glycerol of phospholipid. Here you can see fatty acids undergo breakdown from the first and third position by the action of enzyme lingual lipase, gastric lipase or pancreatic lipase. After its action, they leads to the formation of monoglyceride, which is nothing but glycerol plus one molecule of fatty acid. And free fatty acids are also formed. In the second picture, you can see the transport of the various digested product in the form of micelle to the intestinal mucosal surface. Here you can see some important lipases. Already we discussed about lingual lipase and gastric lipase. Lingual lipase and gastric lipase acts in mouth and stomach. Particularly it acts on triacylglycerol with short chain and medium chain fatty acid. Pancreatic lipase acts in small intestine. It acts on triacylglycerol with long chain fatty acid. For the action of pancreatic lipase, there is a requirement of colipase. It is an enzyme which is secreted from the pancreas in inactive form as a zymogen procolipase. This procolipase is converted into colipase by trypsin. This colipase enzyme binds with lipid water interface, and because of that it makes it possible for the pancreatic lipase to get anchor to the lipid water interface and act on triacylglycerol and causes its breakdown. Intestinal lipase also acts in small intestine along with bile acid. It causes breakdown of triacylglycerol with medium chain fatty acid. Phospholipase A2, as we already discussed, it causes release of unsaturated fatty acids from the position 2. Lipoprotein lipase and hormone sensitive lipase, these are two most important enzymes. Lipoprotein lipase is secreted by the endothelial cells of capillary wall. This enzyme is essential for the breakdown of triacylglycerol present in chylomicron and VLDL. This triacylglycerol present in chylomicron and VLDL, when undergo breakdown, it forms free fatty acid and glycerol. This free fatty acid and glycerol is later on internalized by various cells of tissue. Hormone sensitive lipase is also important enzyme which is stimulated by noradrenaline and glucagon. This hormone acts in adipose tissue. It acts on triacylglycerol stored in adipose tissue and causes its breakdown into free fatty acid. This free fatty acid is later on utilized for beta oxidation of fatty acid. Now we can see the role of bile in fat digestion and absorption. Bile are biological detergents synthesized by liver and secreted into the intestine. Bile secretion is composed of bile acid. Bile acids are nothing but cholic acid and chenodeoxycholic acid. These bile acids are synthesized from cholesterol precursor. When these bile acids are conjugated with sodium and potassium salts, they are considered as bile salt. Bile acid and bile salt together play an important role in emulsification and micelle formation. This is possible because of the peculiar structure of bile acid. Bile acid is having two portions, hydrophilic side and hydrophobic side. Hydrophilic side is water loving side and hydrophobic is water hating side. Bile acid or bile salt get arranged themselves in such a way that it forms a layer around fat droplet or lipid droplet. Here, water loving hydrophilic side is facing towards exterior and water hating hydrophobic side is facing towards the internal side. Because of this, lipid becomes somewhat soluble in aqueous medium, and because of this type of formation, these lipids can undergo digestion and absorption very easily. You can see the role of bile in emulsification and micelle formation here. When we pour oil into aqueous medium, oil particles are dispersed into fat globules. These fat globules later on coalesce with each other and form fat surface at the top and water medium at the down. Fat is occupied at the top level because the density of the fat is lesser than water. This aggregation of fat globules to each other is prevented by bile. Bile reduces surface tension of fat globules and make themselves arranged in such a way that there is a prevention of aggregation of fat globules. These fat globules are arranged in micelle formation, and because of that there leads to increase in surface area of the fat, which is essential for fat digestion and absorption. In the intestinal lumen, you can see here fat globules undergo emulsification process by bile salts and form emulsion droplets. These emulsion droplets later on undergo digestion by various lipases and form digestive product and bile salts. These bile salts again recombine with digestive product to form micelles. These micelles are responsible for the transport of digestive product from intestinal lumen to the plasma membrane of the mucosal cells of intestine. At the mucosal cell surface, micelles release digestive product which later on pass through the plasma membrane of mucosal cell into the intestinal cell by diffusion process, and bile salts are remain in the intestinal lumen, which later on undergo enterohepatic circulation and goes into the liver and again re-secreted. In this diagram you can see the same. Large fat droplets undergo emulsification by bile acid, which later on undergo digestion by various digestive enzymes, and digestive products are formed, particularly monoglycerides, fatty acids, cholesterol and lysolecithin. These digestive products later on internalized by intestinal mucosal cell by diffusion process. Monoglyceride and fatty acids together combine to form triglycerides, and these triglycerides along with cholesterol and phospholipid, which is resynthesized again, get incorporated into chylomicron, and this chylomicron is exocytosed in lymph. So for lipid digestion and absorption there are total 5 different phases: hydrolysis of triglyceride to free fatty acid and monoacyl glycerol, solubilization of free fatty acid and monoacyl glycerol by detergent, bile acid, and transportation from intestinal lumen towards the cell surface, uptake of free fatty acid and monoacyl glycerol into the cell and resynthesis to triglyceride, packaging of triglycerides into chylomicron, and exocytosis of chylomicron into after absorption of lipid into intestine and its packaging into chylomicron. Now we'll see secretion of lipids from intestinal cell. This will occur with the help of lipoprotein particle, which transport lipids from intestine for absorption and utilization by the cell. Lipoprotein particle, particularly chylomicron, is produced in intestine and it is responsible for transport of lipids from a meal. This chylomicron particle is composed of fatty acids surrounded by phospholipids and protein and soluble in water. You can see here phospholipids, cholesterol, long chain free fatty acids and monoglycerides. These molecules are incorporated into chylomicron in endoplasmic reticulum of the intestinal cell, and from there chylomicron is released into the lymphatic system, and from lymphatic system via thoracic duct it enters bloodstream. Medium chain and short chain fatty acids, which are absorbed into the intestinal cell, will enter into the portal circulation directly without incorporation into chylomicron. This is the structure of chylomicron particle. You can see protein and phospholipid at the periphery, and the core is occupied by cholesterol ester, free cholesterol, triglyceride. Now we can see the absorption of fat into various cells. Triglyceride in the chylomicron must be disassembled by lipoprotein lipase before they can enter body cell. In body cells, triglycerides can be used immediately for energy, and or it can be used to make lipid containing compounds, or it can be stored in muscle and adipose tissue. Now we can see the clinical significance of lipid digestion and absorption. There are various conditions which can lead to abnormality like pancreatic insufficiency, which may be due to acute or chronic pancreatitis, and cystic fibrosis. Deficiency of bile salt, which may occur in ileum resection. Ileum resection leads to deficiency of bile salt because ileum is the site which is responsible for reabsorption of bile and its intrahepatic circulation and resecretion from the hepatic cells. So, deficiency of bile salts will occur in ileum resection. Bacterial overgrowth causes deconjugation of bile salts. Decrease in intestinal cells for absorption is also one of the reasons for abnormality. Failure of synthesis of apoprotein, particularly beta apoprotein, can lead to deficient chylomicron formation. For chylomicron formation, B48 apoprotein is required, and if this B apo48 protein is deficient, there is decreased formation or no formation of chylomicron, and so because of that, lipids which are absorbed into the intestinal cells, they are not transported into the lymphatic system and later on to the blood. Abnormality of lipid digestion and absorption leads to one of the conditions known as steatorrhea. Steatorrhea is a condition characterized by loss of lipid in the feces. This occurs due to defect in the secretion of bile or pancreatic juice into the intestine, or impairment of lipid absorption by the intestinal cells. There is a difference between steatorrhea of pancreatic juice deficiency or defect in the secretion of bile. Whenever there is a defect in pancreatic juice secretion, fat which is excreted into the feces is unbroken fat, whereas in case of defect in the secretion of bile, the fat which is excreted into the feces is broken fat. Next condition is cholesterol stone or gallbladder stone. Cholesterol stone or gallbladder stones are more common in female. Obesity is one of the risk factors for cholesterol stone formation. Cholesterol stones are formed when liver secretes bile which is supersaturated with respect to cholesterol. Because of cholesterol stone formation, there may lead to obstruction to bile flow, which leads to impaired fat digestion and absorption. Treatment for cholesterol or gallbladder stone is given by chenodeoxycholic acid. Chenodeoxycholic acid is responsible for dissolution of the cholesterol stone. If this treatment fails, ultimate treatment is given by surgery. Surgery removes cholesterol stone, or it may remove gallbladder also. Next condition is obesity. What is obesity? Obesity is nothing but excessive deposition of fat in adipose tissue. When we consume excess fat in the diet, that leads to excessive digestion and absorption, and in turn excessive deposition of fat in adipose tissue. To treat this obesity, there is a drug known as orlistat. It is a structural analog of triacylglycerol, but it is a non-hydrolyzable substance. It is a powerful inhibitor of pancreatic lipase and is used to prevent fat digestion and absorption. Apart from orlistat, there is another drug known as olestra. Olestra is nothing but synthetic lipid. It is produced by esterification of natural fatty acid with sucrose. Here, instead of glycerol, sucrose is used for esterification with fatty acid. This synthetic lipid tastes like natural lipid, but it cannot be hydrolyzed and therefore excreted unchanged. So, olestra and orlistat, these are the two medications which are used for the treatment of obesity.